Thank you for this very kind introduction. And I must confess, I've been looking forward to coming here because we share a lot of interests in the field of glial biology and also in the field of aquaporin, aquaporin specifically. So what I'm going to do in this lecture is simply to try to retrace some of our steps into this field, starting with the first studies that we did quite some time ago and ending up with some of the unpublished data that we think are beginning to shed some light on the physiological roles of aquaporin in the brain. Because that's the big challenge. One thing is what Aquaporin-4 does in terms of pathology, but when it comes to the normal physiology, it's a pretty difficult task to unravel the specific roles of Aquaporin-4. So, as you all know, the Aquaporin-4 channel is a fantastic molecular machine that lets through water quite specifically. And the reason why the Aquaporin-4 channel is so specific, of course, is that there are specializations along the pore, including a size restriction about here, and also some side chains, so amino acids sticking into the pore, that keep all other molecules than water out from the pore. So even protons are not allowed to enter the pore, which is a good thing because otherwise, of course, the aquaporins would have messed up our pH regulatory mechanisms. So the work that I'm going to tell you about today is in fact more or less exclusively focused on Aquaporin-4. And there are two major players in my group that work on Aquaporin-4, and these are two very talented postdocs, Mahmoud Hamidi Moghadam and Alan Nagelhuus. So most of the work that I'm going to describe to you today has in fact been performed by these two postdocs and their students. And as Christian pointed out, a large part of the group is still involved in research on glutamate. So that was the original interest of our group. But the reason why we switched also to some work on aquaporins is that we believe that, for instance, in stroke, there is no way you can achieve cytoprotection without also having some sort of attention to the edema that is being formed after a stroke. So the reason why we're working on edema and on excitotoxicity with glutamate is that we think that these two mechanisms are so closely interlinked that you cannot really understand one of these mechanisms without also understanding the other. So there is a vicious cycle, we believe, that is being set up, for instance, in a stroke that involves cell death, edema, leading to more cell death and even more edema and so forth. So even though aquaporins are quite different from the molecules involved in glutamate neurotransmission, there is a very, very strong pathophysiological coupling here, which in fact was the factor that brought us into this field. So many of my colleagues, they question our interest in water, because what can be more boring than water? So I have to tell them then that water is, of course, a very common molecule in our body. It's a bit sad to think about it, but more than 90% of the molecules in our body are in fact water molecules. So many diseases are characterized by deficiencies in water transport or water redistribution, and this is some of the conditions that are listed here. And obviously stroke is by far the most common and most important conditions that is coupled to problems with the water transport and water distribution. First of all, back to basics. How are aquaporins expressed in the brain? And I think you know most of the details already, but I will very quickly go through the basic ideas here. The brain contains at least 3 functional aquaporins. I will not at all deal with aquaporin 9. We have recently published a study on aquaporin 9 in the brain, so if you're interested, you can go to that particular paper. But in this context, I will, as I say, focus on Aquaporin-4, but I should remind you that also Aquaporin-1, of course, is expressed in the brain and has a very important role in as much as Aquaporin-1 is expressed in the choroid plexus. There is no doubt that aquaporin 1 is involved in the secretion of cerebrospinal fluid. But aquaporin 4 is the predominant aquaporin in the brain, and it is expressed in 3 different modes. First of all, aquaporin 4 is expressed in ependymal cells, also in osmosensitive structures like the supraoptic nucleus, where we believe it's involved in osmotransduction. But the expression mode that is most relevant for the topic of today is the one that you see here. Aquaporin 4 is enriched in the astrocytic end feet that abut on the microvessels of the brain. So here aquaporin 4 occurs in very high concentrations, as you see in the immunogold preparation here. This is an endothelial cell. You have the basal lamina, an astrocytic end foot, and the immunogold particles signify the presence of aquaporin 4. And also of relevance to what I will be discussing later on is the fact that aquaporin 4 was very soon found to be co-expressed with the potassium channel KIR4-1. And this was a study that Alan Nagelhus did quite some time ago, and he simply quantified the expression density of aquaporin 4 and KIR4-1 in different membrane domains of Müller cells in the retina. So this is a Müller cell. You can see the end feet, and you can also see the microvilli that extend into the subretinal space. So this was what he found. All the membrane domains that contain a lot of aquaporin 4, they also express high amounts of KIR4-1, and vice versa. Plasma membrane domains with little aquaporin 4 also show modest levels of KIR4-1. There's only one notable exception here, and that is that in the initial part, the basal part of the microvilli, there is KIR4-1 but no aquaporin 4. Otherwise, there is a perfect co-expression. And the functional implication of this, we will get back to later. This is again how it looks like in the electron microscope. And for those of you who are interested in anatomy, you should note what is very conspicuous here. And that is that aquaporin 4 is found in the membrane domain that is coupled to the basal lamina here. But at the very instance when this membrane is reflected away from the basal lamina and goes down here, it loses more or less its entire content of aquaporin 4. So we initially assumed that there must be something here in the basal lamina that keeps aquaporin 4 in place, and I will come back to that later as well. And the enormous enrichment of aquaporin 4 in the end feet membrane is perhaps, or most likely, the result of an induction process, because if we make co-cultures of endothelial cells and astrocytes, these are astrocytes, green is GFAP, red is aquaporin 4, so you see aquaporin 4 in the membranes. So if we make a co-culture between astrocytes and different cell types, we find that if we co-culture astrocytes with brain endothelial cells, which is done here, you have an enormous density of aquaporin 4 in the membranes. So brain endothelial cells somehow induce this enormous expression of aquaporin 4 in the perivascular astrocytes membrane. So the other data points here simply show that other endothelial cells and other molecules do not really serve to induce the expression of aquaporin 4. And I should also point out, and this is important also for the physiological data to come, that aquaporin 4 is expressed also in the astrocytic membranes that surround excitatory synapses. It's difficult to see here perhaps, but these are astrocytic membranes. This is a parallel fiber spine synapse in the cerebellum, and there is a modest expression of aquaporin 4 also in this membrane domain. And this is large magnification, a spine, parallel fiber synapse, and you see gold particles indicating aquaporin 4 in perisynaptic astrocyte membranes. So this is reflecting what is becoming more and more apparent, that astrocytes are extremely polarized when it comes to their function and their expression of different proteins. So we compared the distribution of aquaporin 4 with the distribution of glutamate transporter. In fact, this is the EAAT1 transporter. And the gold particles, the large ones, show this glutamate transporter, and the small particles, the small particles show the glutamate transporter, and large particles Aquaporin 4. And there is an inverse relationship here. So the end-foot membranes contain a lot of Aquaporin 4, low amounts of the glutamate transporter, and the exact opposite is the case for the perisynaptic membranes that you see around the synapses here. It's a bit light in the room, but you should trust me that this is the case. So then what we wanted to do next was to try to understand what is the role specifically of the Aquaporin 4 pool that is expressed in the perivascular membrane domain of the astrocytes. And of course, then we needed to knock out selectively this particular pool. So we didn't go for a general Aquaporin 4 knockout. We have this made such a general knockout as well. But specifically, we wanted to knock out Aquaporin 4 just in this particular membrane domain. And please be reminded that the density here of Aquaporin 4 in these membrane domains is excessive. So if you look in a freeze fracture preparation, you will easily see these square arrays that are composed merely of Aquaporin 4 molecules. So it's really a very, very abundant molecule in this particular membrane domain. So what was the first step then? The first step in order to achieve a membrane domain specific knockout was, of course, to understand how is Aquaporin 4 anchored in the perivascular astrocyte membrane. And as you remember, I pointed out that the membrane domain that contains Aquaporin 4 in large amounts faces the basal lamina. So at the outset, we assumed that there must be a link to the basal lamina somehow. And here is the basal lamina. So this is simply a cartoon showing at large magnification what goes on in this area here. And thanks to a collaboration with John Neely in Peter Agger's lab, what transpired was that we could identify as the predominant anchoring mechanism the dystrophin complex. DP71 is the brain version of dystrophin. And the dystrophin complex contains several associated protein molecules, including alpha-syntrophin. So thanks to this collaboration with John Neely, we could find that alpha-syntrophin is coupling Aquaporin 4 to this particular end-foot membrane. So the link to basal lamina and to laminin in the basal lamina is provided through this molecular bridge that is composed of beta-dystroglycan and alpha-dystroglycan. So this link to laminin explains why Aquaporin 4 is concentrated so specifically in this membrane domain. So the prediction then was that if we could knock out alpha-syntrophin, then we could remove Aquaporin 4 selectively from this perivascular membrane so as to assess the function of this molecule in this particular location. And interestingly enough, at this particular time point, Stan Frunner in the University of Washington, Seattle, had constructed an alpha-syntrophin knockout, which he provided to us. So this was sheer luck, of course, but sheer luck is the most important ingredient of science. So we got these animals, the alpha-syntrophin knockout animals, and we were very excited because now we hope to see that Aquaporin 4 was removed from the perivascular membrane. But the disappointment was immense because when we looked at the light microscopic level, there was no change whatsoever. No change. And why was there no change at the light microscopic level? Because the changes were so subtle that we needed an electron microscope to see them. So this is what we saw. It's a bit difficult because of the light, but perhaps you can see the situation here in the wild type situation. Here is the endothelial cell, the capillary lumen, the end foot, and these are the gold particles again showing Aquaporin 4. And in the alpha-syntrophin knockout, there is almost no remaining gold particle here. So the experiment worked. The perivascular pool of Aquaporin 4 was more or less eradicated. But why didn't we see any change at the LM level? Well, that's because in this membrane domain, the other side, so to speak, of the end foot, very close by, there was still Aquaporin 4 present and even more Aquaporin 4 present than in the wild type. So at the LM level, we simply couldn't differentiate between these two membranes, which are very, very closely opposed. You know, the end foot is pretty thin. So this shows the importance, I'm glad to say, of the electron microscope. It's still a technique that is important to use in certain contexts. So, armed with this particular model, we could address this very simple hypothesis that this pool of Aquaporin 4 is of importance for letting water into the brain when an edema is being formed. So the crucial experiment, it was published quite some time ago now, was this one, the MCAO model. You have the normal situation here, tremendous damage to the affected hemisphere, as you see here, edema and cell death. But in the alpha-syntrophin animals, where the perivascular pool of Aquaporin 4 is removed, there is protection. So the size of edema and cell death is reduced by at least 50%. And this is not only true for edema after stroke. We tested several other edema models, and the same thing holds true in all cases, that removal of Aquaporin 4 offers protection. So this is an MR imaging study, apparent diffusion coefficient, and what you see is that the onset of edema is moved from here to here, that is delayed in animals with alpha-syntrophin knockout. So also in hyponatremic edema, is there a protective effect of removing Aquaporin 4? So we were, of course, criticized because in such a knockout, showing an effect, you don't necessarily conclude, or it's not obvious that there is this coupling between the removal of Aquaporin 4 and the protective effect when it comes to the generation of edema. So what some referees asked us to do was to really show that when an edema is being formed, water first enters the astrocytes. And you know, this is not an easy task, because it cannot be done in cultures, obviously. It has to be done in the in vivo situation. So what we started to do was to examine this with multiphoton microscopy. And of course, you have a setup here so everybody knows the technique. So this is our mouse. And what we set out to do was to see if, in fact, the astrocytes are the first cell to swell when an edema is being formed. Because this is what you would predict if Aquaporin 4 is crucial for letting water into the brain when an edema is being generated. And these are some of the data. What Gabriele Nase did was simply to identify astrocytes in different parts of the brain, including the cortex and the cerebellum. And she monitored the volume of these astrocytes in the first time period after exposing these animals to excess water. So this is a very simple model. We simply inject water in the peritoneal cavity, and this induces, of course, a hyponatremic edema. And some of the astrocytes then show a tremendous increase in volume after this hyposmotic stress, like the one shown here. And this one shown in red is the one here that is very close to a vessel. Astrocytes that are more removed from the vessels, they seem to keep their volume more constant. So there is a heterogeneity here among the different astrocytes that you see. And of course, we have not really broken this down to specific astrocyte subgroups, so the only classification we have is the distance to the vessel. But these data indicate that astrocytes indeed are primary sites of water entry, which fits with the idea that these cells contain quite a lot of Aquaporin 4. So more recently, and I don't want to show this here, we are now trying to develop inhibitors for Aquaporin 4. And just a few days ago, a paper was published on the synthesis of ligands that fit into one of these binding sites on Aquaporin 4. So we are pretty optimistic that we will be able to generate an inhibitor within the next 20 years or so. So then switching to the physiological roles of aquaporin 4, which is really a very challenging task. And I think that Christian would subscribe to this statement that surprisingly enough, we still don't know exactly what aquaporin 4 is doing in the normal brain. We have some hypotheses, we have some data, and I will discuss these with you, but the final conclusion as to the physiological role of aquaporin 4 is still not within sight. So luckily we have still some more to do. But the thing is that the first hypothesis that we came up with in regard to the role of aquaporin 4 was that aquaporin 4 was somehow involved in the removal of potassium, in the clearance of potassium. And the reason was simply the one that I already showed you, that KIR4-1 and aquaporin 4 are strictly colocalized, at least in some populations of astrocytes, including the Miller cells. So what is the hallmark, of course, of potassium clearance is that those potassium channels that are involved in potassium clearance, they show this accumulation and enrichment in the end-foot membranes. And this was described a long time ago, of course, by Newman and others, that most of the potassium conductance is concentrated in the end feet. And as I showed you, the same thing holds true for aquaporin. It is concentrated in the end feet. So the hypothesis that we formulated already almost 10 years ago was that effective clearance of potassium may depend on the presence of water channels and may require that the expression of aquaporin 4 be tuned to the potassium fluxing capacity. This we wrote long before we had any experimental data except the anatomical data to support this suggestion. Another hypothesis that I will come back to, and this is unpublished material, is that in addition to be involved in potassium clearance, aquaporin 4 might be involved in osmosensation together with another molecule, and this molecule is Trpv4, and I will come back to that later. And then please be reminded that aquaporin 4 is not only found in the end feet surrounding vessels, but also in the end feet that abut on the subarachnoidal space, and also the Virchow-Robin space that simply is an extension of the subarachnoidal space. So I will come back to this later. We think that here aquaporin 4 is involved in a very important process, osmosensation coupled to regulatory volume decrease, because the subarachnoidal space is a sink for water, for instance, at the onset of edema. I will come back to that later. So first, the potassium link. This is again this colocalization of KIR4-1, large particles, and aquaporin 4, small particles in the end feet. And this shows the general idea that potassium is being taken up from the synaptic areas and siphoned through the end feet. And as will be clear later, we think that this potassium flux is associated with a water flux, and that there is an obligatory coupling between the two kinds of flux. And one of the functional experiments that we did quite some time ago together with Otto Witte was to show that if you stimulate the afferents to a cortical slice, so this is a stimulation electrode down here, it's difficult to see. These are the layers of the cortex, dark field. When you stimulate with an electrode down here, you get a shrinkage of this extracellular space in the target area of these fibers. And an extension or an enhancement of the extracellular space in the more superficial layers of the cortex. So what we think is that when you stimulate here, or the afferents of this area, you set up a water flux from deeper to more superficial parts of the cortex. And with potassium-sensitive microelectrodes, we could then show that exactly the same thing happens with potassium. There is a shift of potassium or a flux of potassium from these deep to superficial layers. So there's a parallel flux of water and potassium. And what we then did was to test this hypothesis of there being a coupling between water and potassium transport in the alpha-syntrophic knockout animals. And this was a very simple test. The prediction would be that depletion of N-foot aquaporin 4 would interrupt potassium siphoning. This would be the case if these two processes were in fact coupled. So, and these are some of the data. This was done in hippocampal slices, stimulation of Schaffer collaterals, potassium-sensitive microelectrodes. What you see, of course, is that the potassium level in the extracellular space is increased during stimulation, and then there is a clearance of potassium as soon as you stop stimulating the collaterals. And in the alpha-syntrophic knockout, you have quite clearly a delayed removal of potassium, as you can see by the overlay here. So we concluded that indeed, in this particular physiological or experimental situation, when you remove aquaporin 4, you also interfere with potassium clearance. And from Workman's lab, soon after, a couple of years or 1 year after, corresponding data appeared with an entirely different methodology, because what Workman did in this case was to develop a marker for potassium in the extracellular space. So he could sort of measure the potassium concentration or changes in potassium concentration simply by a dye that becomes fluorescent when it couples with potassium. And using this methodology and spreading depression, he could show then that in aquaporin 4 knockouts, there is again a reduced potassium clearance or less efficient potassium clearance when you remove aquaporin 4. And Binder did a similar experiment. I know that Binder has been collaborating also with this environment here. So what he did was to stimulate the cortex at high frequencies and show that in aquaporin 4 knockout animals, again, there is a reduction in the efficiency by which potassium is cleared after stimulation. So several lines of evidence do support the idea that there is a coupling here between water transport and potassium transport. So the conclusion is that the fact that aquaporin 4 minus animals mimic alpha-syntrophin minus animals in regard to delayed potassium clearance points to the involvement of aquaporin 4 in this process. If aquaporin 4 were of importance for potassium clearance, you would expect that removal of potassium also would increase the severity of seizures in experimental models of epilepsy. And one very simple model is the one that we used here, the hypothermia model, where the mice experience seizures after an elevation of body temperature. And in this very simple experiment, we could then show that indeed mice that have been subjected to an alpha-syntrophin knockout and thus lack perivascular aquaporin 4, they go into more severe seizures than the wild-type counterparts. And we were motivated by these studies to look at tissue surgically removed from patients with temporal lobe epilepsy. And we were very surprised then to find that indeed in patients with mesial temporal lobe epilepsy, there is a decreased aquaporin 4 level around the vessel. So this is the normal situation, control tissue, strong aquaporin 4 labeling around the vessels. This is light microscopy. And this is what happens in the patients. There is a reduced level of aquaporin 4 in the perivascular membranes. This could be confirmed also by electron microscopy. But the total level is not changed. You can see now there is more aquaporin 4 in the tissue or in the neuropil, so that there is a lack of polarization. The total level doesn't change. There are many ways to explain this finding, and we have been discussing some of these different explanations previously today. But it is a note that in these very same patients, the entire dystrophin complex that is responsible for anchoring aquaporin 4 is missing from the perivascular membranes. This is the control DP71 antibody, as you would expect, very strong labeling in the perivascular area. And in the patients, there is virtually no DP71 labeling. So this suggests that the entire anchoring complex is lost from this particular membrane domain. Or, of course, there could be other cells that have taken the place, so to speak, of the original astrocytes. And interestingly enough, we just published a paper together with a group in Oslo, where we were interested in the connection between MMP9, matrix metalloproteinase 9, and epilepsy. And the reason why we're interested in this is, of course, that this link here between the dystrophin complex and laminin is very sensitive to metalloproteinases. So one of the targets or substrates of MMP9 is, in fact, beta-dystroglycan and alpha-dystroglycan. And MMP9 is an enzyme that is found in the extracellular space and is increased under certain pathophysiological conditions. So one of the theories that we have now is that the reason why the dystrophin complex is lost in epilepsy, and also, in fact, after ischemia, is that you have an activation here of MMP9. This is still a theory, but I think it's quite clear that now the data supporting this theory are increasing in strength. So it is of interest then to look into the genetics here. So we have a rather large cohort of patients with temporal lobe epilepsy, and we have looked into SNP patterns in these patients. And in fact, there are some SNPs that are specifically coupled to temporal lobe epilepsy. And the significance is pretty high. These are data that haven't been published as yet. Unfortunately, the SNPs that are of interest here, they are not in the coding region of the Aquaporin 4 gene. So we still don't know exactly whether there is any functional link between these SNPs and the epilepsy. So this is something that has to be worked out. Unfortunately, we don't know enough, for instance, about the promoter region for the Aquaporin 4 gene. So the last hypothesis that I will discuss with you is very new data, based on very new data, that suggests that Aquaporin 4 might be involved in osmosensation together with another molecule, Trpv4, and also then by inference in RVD, regulatory volume decrease, which is one of the protective measures that cells use to guard themselves against detrimental changes in volume. If you look back in phylogeny, one of the biggest challenges any organism faces is changes in the external environment when it comes to osmotic gradients. So all organisms, be these unicellular organisms or eukaryotic organisms, they have to have ways by which they can protect their volume against inadvertent changes in the concentration of osmolytes in the external environment. So the hypothesis is that Aquaporin 4 forms an osmosensing complex with Trpv4 in the subpial astrocyte membranes. So what is Trpv4? I guess that most of you know this molecule. It's a very interesting molecule. It's a calcium permeable cation channel that is gated by a number of stimuli, including cell swelling. And it's known from other tissues that it acts as an osmosensor, for instance, in the kidney. And what was very striking, and these anatomical data have been published, was that Trpv4 is present in astrocytes, in cultured astrocytes, in quite high amounts. And when you go into the function of the Trpv4 in these astrocytes, you can easily see that if you apply a known agonist of this Trpv4 molecule, you obtain an intracellular calcium increase. And this depends on calcium influx, because if you remove calcium from the medium, you don't see this calcium signal any longer. So when we look into the intact brain, what we find is that Aquaporin 4 and Trpv4 here in red. Again, it's a bit too light to see this difference. But the point is that Aquaporin 4 and TRPV4 are co-expressed at the surface of the brain and also surrounding the Kirchhoff-Robin spaces. And co-immunoprecipitation experiments suggest that these two molecules in fact form a multimolecular complex. And this can be shown both ways. Here we have made an immunoprecipitation with Aquaporin 4 antibodies, and you can find TRPV4 immunoactivity in the precipitate and vice versa with TRPV4 antibodies. And this reflects a true interaction because we don't see this co-immunoprecipitation if we express the two molecules in different cells and just mix the cells. And then we did a very crucial experiment. This was Valentina Benfenati, who was working in our lab, who did this experiment, because we looked at the cell line DINTC cells that contain TRP4 endogenously, but not Aquaporin 4. So we could look at two different situations, TRPV4 alone and TRPV4 in the presence of Aquaporin 4. So this is what happened if you just looked at the DINTC cells without Aquaporin 4 expressed in them. And of course, then you can elicit this calcium signal if you apply 4-alpha-PDD, the agonist of TRPV4. But these cells, having only TRPV4, that do not respond with a calcium signal to hypotonic stress. You see that there is no calcium signal here. But if you co-express with TRPV4, either M23 or M1, the two most common isoforms of Aquaporin 4, you will see that now these cells respond to hypotonic stress. And again, this depends on the calcium influx from the medium. So these data that I showed you here, together with other data that I cannot show because of time constraints, they do suggest that Aquaporin 4 sort of interacts with this TRPV4 channel in osmosensation and also in fact, regulatory volume decrease. The latter data showing that the regulatory volume decrease is also involved here. These data I have no time to show now. So the conclusions are, when it comes to the physiological function of Aquaporin 4, that efficient clearance of potassium from the synaptic region depends on the presence of water channels and also may require, as we have been discussing, that the expression of Aquaporin 4 be tuned to the potassium fluxing capacity. And again, it's clear that there is in the end feed a high concentration of water channels, and these channels might in fact be of importance in the regulation of volume, in the regulatory volume control acting in concert with TRPV4. So these were two hypotheses as to what Aquaporin 4 is doing in the normal brain, but what I haven't talked about is that we also have tried to model the function of Aquaporin 4 in the regulation of extracellular space volume during synaptic activity. And this paper was just published a week ago in PLOS, the PLOS journal. And you could consult this if you are interested in the modeling aspects here. Basically, what this paper shows is that we still don't really know what Aquaporin 4 is doing when it comes to the acute control of extracellular space volume during neuronal activity. We have to be a bit modest here. So these are the people that were involved, or some of the people that were involved. And as you can see, this picture is from Norway. It could equally well have been from Bonn, I guess, taken during the last few weeks. But this is from Norway. This is how it looks like more or less today. So please visit. It's beautiful in the winter, as well as in the summer, of course. So the people involved were many, but I would point out again John Neely. He was instrumental in showing how Aquaporin 4 was anchored through the dystrophin complex. Peter Agre has been involved in many of these studies. I told you about Otto Witte's contribution. Tore Eid used to work in my lab a long time ago. Now he is working in the Spencer lab, and he was instrumental in providing material for the studies of the human material. Anne Williamson has done quite a lot on the electrophysiology. Stan Fröner, as I told you, was the one who constructed the knockout for alpha-syntrophin together with Marv Adams, also working in his lab. And I did not mention these two people, but they have done a lot when it comes to the effect of Aquaporin 4 knockout on the physiology. I didn't go into that. And finally, the Trpv4 story was done in collaboration with Valentina Benfenati and her mentor, Stefano Ferroni. Thank you for your attention.